Hi, my name is Gloria. Today, we shall be discussing the topic biotechnology and genetic engineering. In Module 1 of this topic, we discussed biotechnology. In this Module 2, a discussion shall be on the topic genetic engineering. Genetic engineering. Objectives of lesson. During the course of the lesson, the students should be able to 1. Define genetic engineering. 2. Highlight steps involved in the production of insulin in industries. 3. Define genetically modified organisms. 4. State at least 3 areas genetic engineering is useful to man. And number 5. Mention at least 3 ethnical concerns of genetic engineering. What is genetic engineering? Genetic engineering is defined as a process of altering the genetic material of an organism by either removing, changing, or inserting genes from another organism. Genetic engineering is also defined as the artificial alteration, changing, or modification of genes in cells of an organism in order to change their genetic material and how they work. Did you get that at all? Alright, let's look at it in this simplified form. Assuming you have an organism, we call it organism A. You have another organism called organism B. Genetic engineering simply means taking the genes from organism A and putting them into organism B in order to modify, change the characteristics, or probably change the genetic composition of organism B. That seems clearer. Good. Genetic engineering involves 1. Insertion of human genes into bacterial genes for the production of insulin to treat the disease condition known as diabetes. 2. Insertion of genes into crop plants to make them resistant to herbicides sprayed on them. 3. Insertion of genes from a bacterium into crop plants such as wheat and maize to produce poison that can kill insect pests. And number 4. Insertion of genes into crop plants to provide additional vitamins such as seen in golden rice plants. Now let's look at the processes involved in the making of human insulin. We shall be discussing the processes in steps. So let's go. Step 1. Human insulin genes are selected from human cells. Now a certain chemical is added to this cell so that DNA are extracted from these cells. Now this insulin DNA is cut into smaller equal length sticky ends by what we call restriction endonucleic enzymes. You can simply call them restriction enzymes. Now the sticky end human insulin genes from the chromosomes are then collected. What did I just explain? The first thing is to locate the human cells. From these human cells, you locate the human insulin genes. Now, these human insulin genes contain DNA, so the DNA needs to be located for insulin, of course. And then these DNA are cut into smaller sticky ends by restriction enzymes, and then they are set aside. Then step 2, from a suitable bacterial cell, loose DNA called plasmids are collected. Okay. Now, after that, the plasmid DNA are cut into smaller equal length sizes with sticky ends by the same restriction enzyme used to code the human insulin genes. This is to ensure that the sticky ends are of equal length and they are able to join together precisely to give to carry out the process involved. Alright, so let's move on to step 3. Now, using ligase enzymes, the sticky end plasmid DNA from step 2 and the human insulin DNA from step 1 are joined together. So the fused plasmid and the human insulin gene, known as recombinant DNA, are then returned back. That's it, they are put back into the bacterial cells. Now, the bacterial cells can then be cultured in a fermenter to reproduce asexually. You know that the purpose of reproduction is multiplication. So step 4, what will happen? A large colony of bacterial cells with genetically engineered human insulin genes are then produced. So these insulin genes can be extracted, or this insulin can be extracted from these bacterial cells. They are then filtered and purified for use. Note that insulin injection is used to treat diabetic patients. Take note of this. Insulin controls blood sugar in the body. Now, people who are not able to regulate blood sugar in their body are said to suffer from a disease condition called diabetes mellitus. Now, cow or pig insulin were used to treat this condition previously, but some people are discovered to be allergic to them. So, the need for genetic engineering. So, with the aid of genetic engineering, human insulin modified from bacterial cells are now used. These insulin are said to be pure and cheap, so they can be produced in large quantity and of course commercially distributed. Now, apart from insulin, other products can be produced, so such as proteins. Now, proteins produced by genetic engineering include: 1. Alpha interferons, which is used to treat hepatitis; beta interferons, which is used to treat sclerosis. Now, this image shows us the processes involved in the formation of insulin or the protein mentioned. The first thing is to locate the human cell. After that, locate either the gene for the particular protein or insulin. After that, extract the DNA of these products from them, and then cut the DNA into smaller sizes by restriction enzymes, set them apart. The next thing is to get a suitable bacterium cell, locate the plasmid DNA, cut the plasmid DNA by using the same restriction enzymes as used in cutting the human DNA, after which the plasmid DNA and the human DNA for either protein or insulin and they join together. Don't forget that is called the recombinant DNA. Then this recombinant DNA is transferred back into bacterial cells, and then the bacterial cell is placed in a fermenter where it can reproduce asexually in order to increase in number. So from these multiple cells, this protein or insulin can then be extracted and used commercially in treating these diseases medically. So let's move on and look at this. Take note, the DNA of the organism that contains the DNA from another organism is known as recombinant DNA. So that means if you get the plasmid DNA and you add the human DNA, so the plasmid DNA is now the recombinant DNA. Also know that the organism receiving the genetic material is said to be genetically modified, or you can describe it as transgenic organism. So using our illustration, organism A and organism B, which one is then the genetically modified organism? Good, organism B, because it has received gene from organism A, and so that gene from organism A must have changed the characteristics of organism B or how it works. So let's look at the meaning of genetically modified organisms. Genetically modified organisms. Genetically modified organisms are organisms that contain the genes of other species of organisms introduced into their genes to alter their characteristics. Genetically modified organisms are also referred to as genetically engineered organisms, or you can call them transgenic organisms. It is an organism that has acquired one or more genes from another organism by artificial means. So it's a man-made thing. It's a man-made intelligence thing. So it's not by the normal reproduction process or the traditional breeding method. No, it's a man-made engineered process. Now, this process is achieved by using recombinant technology, and so it can be done in both plants and animals. Why are genetically modified organisms important? Some people say, or some people believe, that these genetically modified organisms are very good. Why? Let's look at some of the reasons. Number one, some farm animals are given engineered genes in order to help improve their egg, meat, and milk production. This makes food abundant in a population. When the egg being produced are in large quantity, the meat in large quantity, enough milk, definitely there will be excess food in a population. It's a good thing, right? Good. For example, we have the dairy cow, which can be given the gene known as recombinant bovine growth hormone, genetically engineered in bacteria in order to increase their milk production. Number 2, farm crops are improved with genes that can improve their nutrients. For example, we have the golden rice mentioned earlier, which is engineered with genes that can help to produce vitamin A. Vitamin A is a good vitamin that gives us nice vision, makes the eyes clearer and we see better. Now this will help to prevent blindness in vitamin A deficient people. Good. Number 3, some farm crops are given genes to make them resistant to herbicides and pesticides. So these farm crops are not affected even if herbicides are sprayed on them. So the weeds, the pests can be sprayed along with these farm crops and the farm crops are not in any way affected. So thus saving time and energy. Number 4, in agriculture, a transgenic embryo containing a desired gene from a third parent can develop successfully, thus increasing animal yield for a particular characteristics desired. For example, you have a fertilized egg from a male and a female organism. It is possible to have some certain characteristics that are not in these two organisms. Now, a third organism gene can then be genetically engineered and then transferred into this embryo, and so you have a better organism in quote. Okay, good. So these are some of the reasons why genetically modified organisms are said to be important. So let's look at the other side. Why are genetically modified organisms said to be bad? Why is it that some people are saying they have ethnical concern, they don't like them, we shouldn't go for them? Let's look at some of the reasons. Okay, genetically modified organisms are of great ethnical concern. So most concerns are centered on genetically modified crops, not on the recombinant microbes themselves. Now, such concerns include 1. Animals genetically modified, they face greater health risks. For example, the dairy cow, because of the recombinant bovine growth hormone modified in cow, they get more infections in their milk duct and are often treated with antibiotics. So humans that eat this cow meat, okay, over the time their body become resistant to any antibiotic they take, because it's like they've eaten a whole lot of these antibiotics, not even taking it, eating it from the meat, and so they become resistant, which is not actually a good thing when someone is actually ill. And number 2 reason is that genetically modified genes may produce products that consumers may be allergic to. You might not know, it might just be there. Okay. Number 3, there is possibility that recombinant DNA technology might create new pathogen that may cause other diseases. Okay, and that is not a good thing. Okay, another ethnical concern is that some crops carrying genes from other species might be hazardous to human health or the environment. And lastly, the use of weed killers such as herbicides and pesticides on genetically modified crops, it will not harm them. Okay, the crops may not be harmful, the pesticide may not be harmful to the crops, but it might be harmful to nearby plants and animals, thereby causing or encouraging environmental issues such as water pollution, air pollution, and eating residuals of these herbicides in these plants, quite harmful. Okay, in conclusion, governments and regulatory agencies throughout the world are working on best ways to facilitate the use of biotechnology in agriculture, because whether we like it or not, it's actually good in some parts, it may be bad in some parts. So they are trying to look for new ways to facilitate the use of biotechnology in agriculture, industry, and even in medicine, while ensuring that new products and procedures are used, okay, in order to make sure that everyone is safe. So finally, we've come to the end of the class. This is what we call the students' activity. So we have 10 questions for you to actually test your understanding of this topic. So it's time for you to pick your pen and your paper and start digging on the question. start digging out the answers from what you heard and you've learned so far. In case you're not able to answer any of them, please don't worry, don't feel bad. All you need to do is go back to the video, listen to it again, view it again, download it for future use, and at the end I believe you should be able to answer all and even more questions on genetic engineering. Before then, if you're a new subscriber, please, new viewer, subscribe to my YouTube channel. If you are new here, please click the notification button, drop your comments, watch the video as many times as possible. Even tell others about the YouTube channel. I think it's actually good for all of us. So at this time I will say thank you actually, but before then we need to mark what we've done. So it's time for you to mark yourself. Our goal here is to have 100%. Our 100% means 10/10. So like I said, thank you for joining me. Have a wonderful time.